Hi, welcome back everyone. Let's continue our discussion of the two regimes that we were discussing in the previous video. So, the regime that we first started with was tau wait or tau occurrence much greater than tau fixation. And in this regime we saw that this point of tau wait is much longer and tau fix is relatively shorter. As a result, when I take a snapshot of this picture, I am much more likely to find the system in this particular state as compared to where two genotypes are coexisting, where this is going to fixation and this is being eliminated, because this process happens relatively faster as compared to the occurrence of mutation. So, that is the first regime that we want to talk about, and that is sort of the snapshot of this regime. There is one more representation of this regime that we should be looking at, because the literature that we read will introduce us to this representation, is the following: that in this particular representation we have on x axis time. So, remember that we are representing exactly this process, but in a different way. Both these are popular representations, and that is why we want to understand what are they actually telling us about the process. So, in this case, this is plotted against time, and on the y axis is number of individuals. What you should realize here is that in the first representation, let us call this A and let us call this B. In the A representation, there is no mention of time in the graphical representation that we are doing. So, graphs have fitness versus number, and they do not have representation of time on them. On the other hand, in the second one, the graph is between time and number of individuals, and there is no mention of fitness per se, although it does make an appearance, as we will see in just a little bit. So, the population size in the chemostat is from 0 to n, and at t equal to 0, this point is t equal to 0, the entire population is isogenic population. So, we start with an isogenic population. All individuals have a fitness of F naught. Now, so we move forward in time. So, on this graph we move from this point to forward in time, and then this time is associated with tau wait, because now the population is waiting for a beneficial mutation to occur. And what is going to happen is that at some point a beneficial mutation is going to take place, and this point represents the fact that there is this one individual which is carrying this beneficial mutation, and the remaining n minus 1 are carrying the ancestral genotype. Now, as we move forward in time, maybe this individual will divide again and its numbers will increase to 2. And what that means is, at this instant of time, I have two individuals of this blue genotype which is of a higher fitness, and n minus 2 individuals which are at lower fitness. Also, the way to read these graphs is that at any given point in time, if I want to find the composition of the population, that how many individuals are there of a particular genotype, what I do is draw this vertical line here from 0 to n, and this vertical line at any given point in time represents, this is the number of blue individuals, and everything from there to n, which is the size of the system in terms of the carrying capacity of the chemostat, everything from this point to n is the number of black individuals that are present in the population. So, if I were to draw that graph here, this graph here simply tells me that all individuals here are of black type and there is no diversity that is available in the population. So, that is how I can measure the relative frequency of the two genotypes, which at this point for black individuals is n minus 1 upon n, and for blue individuals that is simply equal to 1 upon n. But remember that the most likely fate of a beneficial mutation is that it is going to go extinct from the system. So, the fate of this population can be represented as simply this. So, beneficial mutation came, it fluctuated around a bit and then it went extinct. This is the process which in the first representation was depicted by this beneficial mutation occurring, but then lost due to drift, and as a result the system going back to the state that we started with. The same is happening here. Initially it was all n individuals, and after the beneficial mutation went extinct, I am back to the state where there is only all n individuals in the system are of the ancestral genotype represented by black circles here. So, the system went back to the state that it was in prior to occurrence of this blue beneficial mutation. And since on an average we saw that 19 mutations, beneficial mutations occur and they are lost, and then out of 20 only one survives, this will keep on happening. So, this fate will keep on happening. Some will persist for a little longer, but then eventually go extinct, some might immediately go extinct, and so on and so forth. So, in this representation we have all this information that can be read if we have that detailed information available from the experiment. Eventually though, what will happen is that finally there will be some beneficial mutation that occurs which is able to survive drift, which roughly will be 1 in 20. So, its numbers go up and down because fate is decided by selection and drift, but its selection is weaker as compared to the strength of drift when numbers are small, but eventually it escapes drift and increases in numbers. Once it increases a certain threshold, now the fate of this population is no longer dictated by drift to the extent that it was when numbers were small. Now onwards, the fate of this mutation is going to be decided primarily by selection. So, this happens when its numbers in the chemostat have gone up to a sufficient degree that it is unlikely that all these individuals carrying this beneficial mutation are going to be eliminated by selection only. If it was 1 or 2, then it is quite possible that these individuals might be lost because of drift, but now there are so many that it is unlikely that all of them will be lost by action of drift without giving selection a chance to increase their frequency. So, what that means is that here on onwards, this increases in frequency more or less deterministically, exactly as the exponential exactly as the equation for two competing genotypes in a chemostat that we derived several videos ago, exactly in accordance with those rules, and this will increase in frequency and eventually reach fixation. And now this process will repeat itself over and over again, and hence we get adaptive change taking place. At this point, once this has reached fixation, if I were to draw a vertical line, so at at this point the frequency of this genotype is n upon n, which means it is just 1. That means this genotype has reached fixation. On the other hand, if I were to ask at this point that what is the frequency of this genotype at this point, that is now 0 upon n, which means this genotype has become extinct. So, one has reached fixation and the ancestral genotype has become extinct. So, now in the representation that we have done here, you should note a couple of things. One is that the time it took for a beneficial mutation to come before it could survived the action of drift is this tau weight. Several other beneficial mutations occurred prior to the one that actually eventually escaped the action of drift, but all these were lost. Tau weight is associated with not just occurrence of a beneficial mutation, but occurrence and being able to survive drift, which happened at this point. So, this was tau weight. From there on, it reached fixation. So, let's call this time in the experiment as T1 and it reached fixation here. So, let's call this time T2. So, in how we have drawn this picture, what we see is that tau weight is equal to T1 and tau fix is equal to T2 minus T1, which is this window of time. And as we can see in how we have drawn this, that tau fix is actually much smaller as compared to tau weight. So, going back to the question that we ended last video on, that if we were to take a snapshot of this picture, if we were to take a snapshot of this chemostat at any given point in time, what would we see? So, if we took a snapshot here, there is no mutation, snapshot here, no mutation, snapshot here, no mutation. If we take a snapshot here, I will get one individual out of n which is carrying this beneficial mutation. But such low frequencies are really hard to detect, because remember the n that we are working with is in hundreds of millions, billions, hundreds of billions, and detecting one individual is really hard in those settings. So, I will not even detect this mutant until it increases to a certain frequency. So, I will not detect it. It is only around here that when I start taking this snapshot that I will see that, oh, this population actually does not have just one genotype, it has two genotypes. So, in this window I will see two genotypes, but rest of the time I am actually only seeing one genotype in this entire chemostat and the time associated with where I saw a mutation go to fixation. So, if we draw, so this is for T2, but if we draw this picture for a much longer time, this graph will begin to look as follows. So, now we do not want to draw it for only T2, but for a much longer time. So, this is 0, t equal to 0, and this is number of individuals, which is 0 to n number of individuals. And now what we will see is that over a much longer time frame, this is what we are going to see. Mutation came, escaped the action of drift and reached fixation. Then we have to wait a really long time for a new mutation to come, which was able to escape the action of drift, and then this mutation also had its fate, and then it was able to reach fixation. And this process keeps on repeating itself, newer mutations keep on occurring and goes to fixation, and so on and so on and so forth. This process will keep on repeating itself. In each one of these, as you will see, let me just fix this. in each one of these, as you will see that the time associated with waiting. So, if we look between red and blue, the time associated with waiting where it escapes drift maybe somewhere here, this is tau wait, and this when it reaches fixation, this is tau fix. And as we can see, in each one of these 3 mutations that fix in the population, tau fix is much smaller as compared to tau wait. We want to discuss 2 other things, that let us compare the ancestral genotype that we started with we will compare this with what we end up with. So, let us say this was the ancestral genotype that we started with. I should also mention that I have not shown in this figure, but what is constantly happening is this beneficial mutations are coming and going extinct because of the action of drift. So, that is constantly happening. In this figure we are only showing beneficial mutations which were able to escape the action of drift. All these little mounts here show beneficial mutations which came, but were quickly lost by the action of drift. So, the first point we want to make is that when we start this experiment, let us say this orange circle represents the genotype of the ancestor. Now, as we move forward to the first mutation, then this has happened, that in the genome of this individual, one red mutation has taken place somewhere. In this little window, prior to occurrence of this red mutation, every individual in the population had this genome. When the mutation occurred, until it reached fixation, in this window coexistence of both these genomes took place, and here on onwards, let us call this time T1, where the first mutation reaches fixation, and here on onwards every individual from T1 onwards, let me write T1 again, from T1 onwards every individual in the population is carrying only this genotype. So, here everyone is carrying this genotype. So, we started with this genotype and this went extinct at T1, right. So, any change that happens after the red mutation reaches fixation is going to take place in this background now and not the ancestral background. At this time, till from T1 to this time, it is only this genotype which exists. From here to, let us call this T2 now, the genotype that exists in the population is the red one, obviously everybody was carrying, but now another blue mutation has happened. So, in this window of time, these two genotypes are coexisting, this one and this genotype which is carrying 2 mutations up until time T2. From time T2 to time T3, in this entire window the only genotype that exists is just this genotype which is carrying both these mutations. This particular genotype went extinct at T2. Remember that this mutation did not go extinct, but that there is no individual left in the population which is carrying only this mutation. Every individual in the population is carrying the red mutation in conjunction with the blue mutation that has occurred. And finally, this process continues, and at T3 a new genotype makes an appearance in the population where we have 3 mutations in that genome. We have the red one from the first mutation that survived drift, and then the blue one, but now a green mutation has also taken place. Let us say this is the green mutation. And eventually beyond this time T4, beyond T4 only this genotype exists. So, as we will see that as an evolution experiment proceeds forward, excuse me, as the evolution experiment proceeds forward in time, we keep accumulating more genotypes on previous genotypes that have reached fixation, like here, like the red mutation reached fixation, and hence newer mutations are going to occur to happen on that background itself, and this is how genomic change will drive itself forward. That is sort of the first point that we want to make, that this is how we can view adaptive change, the genetic changes associated with adaptation. This is how we should be viewing this process. All of this, remember, is still in the regime where we have tau weight much greater than tau fix. The second thing we want to ask is the following, that in this context, now let us zoom in a little bit and talk about a smaller time scale, that what is happening is that beneficial mutations are coming but going extinct, going extinct, and eventually one is able to escape the action of drift, and then selection, more or less selection dictates its fate, which means what we are saying. So, again this is time and this is number of individuals going from 0 to n, going from 0 to n. So, what we are saying that there seems to be a threshold here, and if the mutant numbers are below this, so let us call this some NC, n critical, and n critical, what it is doing, if number of mutants is less than NC, that means these are scenarios where number of mutants are less than NC, then fate of mutation is largely dictated by, and you should all be able to guess by this time that this should be dictated by drift. As we can see here, that we saw that because of the action of drift, even this fit genotype went to extinction. Same here, mutant came which was actually beneficial, but it still went extinct because of the action of drift. Selection would never permit it. But it seems like there is a critical number NC, that if the number of mutants go beyond NC, then in that case the fate of the mutation is now largely dictated by selection. Selection operates via precise mathematical laws. We derived all those equations. So, this trajectory is a smooth trajectory, smooth deterministic trajectory. We can write equations for them, get the exact profile, and every time we do an experiment we will get the same precise trajectory. However, when for mutations whose action is decided by drift, these are driven by chance events. A lot of noise drives these processes, and as a result these trajectories are stochastic. We in this diagram have pictures of 3 beneficial mutations occurring. Let us imagine that the ancestral genotype that we started with, its fitness was F naught, and this beneficial mutation conferred a mutation F naught plus S naught. This one also conferred the same benefit, and this one also conferred the exact same benefit. However, we see that the fate of all 3 is a unique trajectory. This one never went up to very high numbers, it went extinct the fastest. This one was able to increase the numbers a little bit more, and this one was able to escape the action of drift and go to selection. So, despite conferring the exact same benefit, these mutations have different adaptive trajectories, and that is because of the stochastic nature of how drift is going to treat these mutations which confer the same benefit. So, the second point we want to note is sort of ask this following question, that what is the number of individuals of a particular genotype beyond which fate of a mutation is decided by drift, is decided by selection, and drift can be ignored thereafter. And as we can see in this trajectory, from some point onwards this trajectory is relatively smooth, like obeying mathematical laws, and this is the deterministic trajectory. This here is the stochastic bit. And people have worked this out mathematically, and what is believed is that if you have a beneficial mutation, so for a beneficial mutation with selection coefficient S naught, then trajectory is deterministic. Trajectory is largely deterministic if its numbers are bigger than 1 by S naught. What that means is that if you have a beneficial mutation occurring which only provides a very small benefit, in that case, in order to go beyond the action of drift, in order for that mutation's fate to be decided by selection, you need a larger number of individuals. However, if you have a beneficial mutation that confers a lot of benefit, in that case you only need a few individuals, and then selection can take over and drive that mutation to fixation. So, this number beyond which a mutation's fate is decided by selection and it has escaped the clutches of drift, that number is a function of what is the selection coefficient, how much benefit is this mutation conferring upon the population, and that decides how much is the critical number of individuals that allows us to make the assumption that trajectories are largely deterministic thereafter. And we will continue this theme in the next video.